very excited to work primarily with the UK Biobank, but I also work with the Mexico City Perspective Study. That's been amazing, and there's just so many smart young people in Mexico working on that data set now, as well as the China Kadoorie Biobank, and really the emerging Our Future Health Biobank, which, if you don't know, will have eventually 5 million people in the UK with complete sort of birth to death medical records, as well as SNP chips imputed on the UK Biobank. We'll talk probably about imputation and sort of ethnicity later. Obviously, there are pros and cons to imputing on the UK Biobank, but I think that's a complex topic that will lead into some discussion. But there's of course also promise there. I'd love this to be sort of as interactive as possible. Obviously these things range a lot in terms of their interactivity, but I have the chat module up in front of my slides for me. Hopefully you can't see it, just so it's not distracting. But if you have any questions, please feel free to just unmute yourself and just interrupt, or please put them in the chat, and I'll try to get to as many questions as possible if there are a lot of them. Radio silence is okay too, but I'll probably check in once in a while just to make sure that you can still hear me. So with that, I'm going to jump in. This presentation will probably be 25, 30 minutes, giving us plenty of time for questions. And if there are none, obviously we can give everybody a little bit of time back. I picked the longest title ever. So long, in fact, that it took 2 slides. But really, at the end, I wanted to talk about why I think sort of AI and really sort of more generally deep learning analysis is going to drive the creation and expansion of future biobank data. So as I mentioned, a lot of this talk is going to be about my work with the UK Biobank, and I think there's something that not this group, but a lot of people sort of out there in the community forget, which is that the biobanks are really driven by people. And honestly, it has been one of the greatest pleasures of my professional life to work with Mark Effingham, Lauren Carson, Rory, Lucy, and others at the UK Biobank. They're wonderful people and really, really smart and dedicated to their mission, as well as dedicated to the mission to get Biobank out to other people. I don't think I need to tell this group about the evolution of the UK Biobank, but just in case, sort of for completion, it's here. I'm going to run through some of the data types in the UK Biobank lightning quick. Talk about updates if you haven't been following them. And one thing sort of DNA Nexus has done, and it's not the be all and end all, but I think sort of DNA Nexus was sort of a first mover in sort of cohort selection in biobank space. And so that's been really nice, to be able to, even as of 3 or 4 years ago, to be able to say, I'm interested in all women between 45 and 60 who have breast cancer and take cod liver oil. Of course, a big differentiator between the UK and the US and other countries, the consumption of cod liver oil. But anyway, so that's been really a cool thing to work with. And in my personal estimation, I think disease subtyping is sort of the next frontier of biomedical science. Obviously, we've been working on it sort of diligently with breast cancer for the last 30 years, but there's obviously hundreds and thousands of other conditions where we could have adequate subtyping. I mean, thinking about major ones, type 2 diabetes, there's probably 8 subtypes. Alzheimer's, there's probably 5, so on and so forth. So I think one of the biggest things that these biobanks are doing is disease subtyping. And that's really what the sort of middle of the talk will be about. Obviously the biggest use case, maybe not obviously, the biggest use case in the UK Biobank is really sort of GWAS, PWAS, PRS, other population genetic studies. One thing DNA Nexus has done well, in my opinion, is to really have wrapped tools that people can bring in to enable GWAS, PWAS, PRS really quickly. And one thing we found that might be of interest to you is that Regenie is really, really more efficient than other GWAS tools for populations of this kind of size. These papers are, of course, a little bit dated. Now, as you know, there's 500,000 whole genome samples in the UK Biobank, but we've been able to sort of keep up with the times and keep putting up webinars about how to do stuff. And I'll talk a little bit about webinars and sort of expert roundtables a little bit later. Um, Proteomics has really taken the world by storm. I mean, as most of you probably heard, as you may know, there's 50,000 Olink samples, available in the UK Biobank, the UKB rep. And soon there will be an additional 600,000 samples, and some of those will be longitudinal time points. I ran a data management for precision medicine conference last year. By the way, all of those recordings are available. And I asked each panel at the end of their session, Scott was on one of them actually. I asked each panel at the end of their session, what data set they would create if there were really no sort of monetary restrictions. And about 50% of all of the participants on all of the panels said longitudinal proteomics data. So that's very exciting. The other thing that's really exciting is imaging. As a sort of genomics and proteomics person by training, it's been really exciting for me to think about how to integrate imaging data. And in fact, we just put NVIDIA Monai on the UK Biobank so that there's a suite of flexible tools that not only sort of imaging data scientists can interact with, in terms of Monai Core and some of the other offerings we've had, but also clinicians can interact with it through 3D Slicer and some other things. And I think there's a lot of promise in terms of large scale public health with OCT style data. Really the idea that, sort of like your optometrist in, say in the United States in a strip mall can have insights into early dementia and that kind of thing and refer people to neurologists, I think is to me very, very exciting from a public health perspective. And really, being able to integrate all this data with the specific cohorts I talked about earlier, I think is a very valuable thing. But, obviously we've started thinking about integration across biobanks. And I think that there's a lot of shops right now selling federation and saying that they can do sort of seamless federation across cloud infrastructure, across HPC. And I'm going to be frank with you. I mean, I think not all of them are sort of misguided. There's a couple that I've been really impressed by, but there's many, I think, sort of data federation snake oil salespeople out there, and it's something to be careful with. But really, if you're a working data scientist these days, I think really the most promising sort of pseudo federation infrastructure is Docker. Really taking, if you have harmonized metadata, then to actually process the underlying data, one can take Docker containers and drop them into various biobanks. And so I personally think it's critical that we teach young researchers how to use Docker during this period before we really get robust federation. I think one of the bigger problems with federation right now, before I go back to Docker, is that a lot of it is API based. But of course, there are major privacy concerns with these databases. So sending data across APIs is really a non-starter. So federation has to be done in a very clever manner. And I think, people like Bonnie Berger at MIT have done amazing sort of groundbreaking work on the mathematics of like how that federation works. But translating that into sort of a productization is a labor that is just starting to emerge. And I'm happy to talk offline about some of the places that I think are doing a reasonable job. Going back to Docker, I was at a major medical institution in the United States with about 60 postdocs in front of me. And I said, well, what do you need to do? And they said, well, we need to integrate all of us and UK Biobank and another biobank. And I said, okay, great. How many of you know how to use Docker? And two of them raised their hands. That to me was of great concern, right? Because again, unless you partner, there's no sort of clear cross-cloud federator apparent. So unless you're partnering with somebody, it's still going to take years to be able to realize that unless you use something like Docker. So we worked with HDR UK to put out a bunch of educational webinars thinking about how to use Docker effectively, how to use Docker containers, this QR code here, how to build Docker containers, and then really sort of the nitty gritty of phenotype harmonization, what that means. And that may be useful to you as you think about sort of which biobanks you'd like your biobank to cross with, working on other things. Yeah, I encourage you to watch these, and please find me if you have any questions. And oh wow, um, oh, hi, Cindy. I did not know you were here. That's great. So, uh, yeah, Cindy points out that Melissa Klein has a nice publication on Biobank Japan data. So that's great. And yeah, the Biobank Japan people, the people in Japan who run it are just amazing, wonderful people. If you've never met Toshiaki, he's great. Okay. So and I'm really excited about sort of integrating real world data, with a lot of these models. One thing we've been able to prototype, is are thinking about not not sort of replacing clinicians, but of course, sort of clinical decision support. And one thing that an amazing collaborator of mine, who's now a PhD student in computer science at Oxford, was able to do was look at ECG and look at features from ECG and really just make a heat map on the ECG, really highlighting the parts that the model thought were abnormal, so that cardiologists could look at it, but then also highlighting really what prioritizing what the number one abnormality was, so that additional machine learning analysis can be done on those ECGs. So this is a bunch of stuff DNA Nexus does. I think I've pretty much talked all over it, so I don't think we need to go into that too much. But one thing I really want to sort of double click on here is disease subtyping, and really how biobanks are really making that a reality, and really helping to treat patients better, which is a thing that I think we are all extremely interested in. So I think we all know, or it's relatively well known in the field, that colorectal cancer is probably something like 4 subtypes. There will be clinical and academic disagreement about this for a long time, but I think we're all sort of starting to coagulate on the idea that there's about 4 subtypes, and I think the 4 have held up relatively well. There are certainly people say that there's a 2A, 2B, that kind of thing, but more or less, I think that's what we've focused around. And so I did a project in the Elixir Biohackathon. And if you're not familiar with that sort of organization, I'd check it out. We spent a lot of time thinking about if you have, sort of a driver mutation and then background mutations, what drugs are going to be most effective for those patients. And that was really helpful and really cool. And then lately, we've been thinking about integrating proteomic data into those types of analysis too. And I just ran a hackathon the last 3 days at Carnegie Mellon, and we were able to think a lot about integrating pQTLs into genome graphs, and that was a cool project. We're also able to think about with proteomics, being able to think about things like transition of clonal hematopoiesis to myeloid neoplasm. So really taking sort of a subset that we think is going to be aggressive, and being able to predict which subsets of clonal hematopoiesis are going to be oncologically deleterious. So yeah, and then thinking about being able to come up with really simple readouts. For example, with the colorectal cancer example I talked about earlier, this is an example of really sort of reducing the search space, reducing the noise, and not doing deep learning with 25,000 inputs, but doing it with 40, which is really cool, because then we can start fundamentally predicting which drugs are going to be uh, going to be effective for these things. And I think, in my opinion, one thing that we need to do as sort of biobank hosts is be pushing to get subtyping specific vocabularies into the biobanks. And I think one thing that clinical diagnostics, as well as as well as pharma companies aren't really aware of is that they can get data into SNOMED, and there are forms for that that they can fill out and work with SNOMED to get their subtyping data. So that's a message. If anybody would like information on how to get data into SNOMED, I'm happy to share that. So we did I think this is a really nice proof of principle with prostate cancer, where we looked at proteomics and genomics. I'd like to talk about that a little bit. So about 1 in 8 men will be diagnosed with prostate cancer, and I think it's hypothesized that if all men live to 150, 100% would be diagnosed with prostate cancer. But it's about 1 in 10 cases will become aggressive, and it's very hard to predict which ones will become aggressive. So it's often overtreated, which has some unfortunate side effects. So really what we decided to do was take a look at some subtypes of prostate cancer. And we were particularly interested in this subtype characterized by an overexpression of SPINK1 and downregulation of CHD1. And so the really nice thing was that, and this just tells you what SPINK1 is, but what we were able to do was create a cohort of SPINK1 overexpressors based on proteomic, and then extract the CHD1 gene from the whole genome sequencing and look at that. And so it was trivial to really create these cohorts, because we have an infrastructure which allows us to look at each protein individually. And this is, again, just in the pilot study. I'm really excited for the 600,000 samples so we can do this with some statistical power. Again, this is really more of a proof of principle. And then, we can use a bunch of population genomic software. Happy to talk about this offline. Create FASTA files, and then create pangenome graphs just to make sure that there's nothing major going on in the in that region of the genome, copies, deletions, et cetera, et cetera. And I think, in my personal opinion, pangenome graphs are probably where we're going. I talked to Valerie Schneider, the head of the Genome Reference Consortium, and I think she feels strongly that the reason that they haven't released GRCH 39 is because really we're sort of moving over to the Human Pangenome Graph Consortium instead of thinking about human genomes as linear. And I think that's really helpful in a number of ways, including really sort of getting diversity into our genomic analyses. But I'll try to keep this pretty high level. And we're able to Dockerize a lot of this. I've talked about Docker a lot, so I'll skip over a bunch of this. And I think I'm somewhat preaching to the choir, but you're able to work with Docker registries. And it's really nice to be able to use some of this complicated software like PGGB from Docker. And the nice thing is we're able to look at I'll just sort of skip to the punchline. We're able to look at CHD1 and find that there's actually no sort of major changes in the graph. So this is actually a graph representation of CHD1, and you can see that really there's no major changes there. So what we know then is there's no major sort of structural changes in that region of the genome. And we can also see that actually there's many more mutations in CHD1, which is actually very interesting. So for some reason that we don't understand, CHD1 is much more mutated in the upregulated SPINK1, and it doesn't seem to be stop codons. It seems to be an abundance of missense mutations, which is particularly interesting. We can contrast that with MAP3K7, a tumor-associated kinase, and we can actually see in the graph that there's some changes, but we don't actually see any more mutations in tumor-associated kinase. So that's really interesting. Whereas, so those slides got switched. I apologize. But what we can see is that there's a lot more missense mutations in the SPINK1 in CHD1 and SPINK1 overexpressors corresponding to this this aggressive prostate cancer than in less SPINK1. So we have a potential sort of mechanism to understand why we might have aggressive prostate cancer. And so theoretically this can lead to a biomarker, right? And this is just a simple prototype study done in one biobank. So to me, that's very exciting. To move that into other biobanks, looking sort of more multi-ethnic biobanks, I think is a really cogent next step. It's very exciting. And as I'll talk about in a minute, it'll be very interesting to see whether sort of these extended haploblocks in the gene region and around the gene are conserved between sort of the UK Biobank, which is, of course, majority white British people, and other biobanks around the world. So that's really exciting. But I think there's an even more exciting thing we can do. And one thing I'm really interested in is not only having sort of the creme de la creme of bioinformaticians be able to look at this kind of data, but anybody with some basic bioinformatics training. And so this is the most exciting part to me. What I'm about to show you is that we can do some really basic sort of blast searches, and then we can do phenotype association. Usually that's a really fancy thing. My friend Ravi Maduri at Million Veterans in the United States has been working hard on making fancy software for that kind of thing. But if I just do a blast search, and I've cut off the IDs just for sort of an overabundance of sort of privacy caution. But what I've done is I've just taken one of these haplotypes of CHD1 that I showed you before in the graph, and I blasted it into all the other ones. So I've come up with sort of a quick and dirty alignment that anybody with sort of an undergrad bioinformatics or computational biology class, or even some self-training could get. But the most exciting thing is that we see that when we align these things, we see a an assortment of this blast alignment with aggressive prostate cancer. And again, so this is a blast search and a couple of bash scripts. Really, any bioinformatician, anybody with any bioinformatics training whatsoever, should be able to do this. So now we can understand the haplotypes of CHD1 that are associated with aggressive prostate cancer. To me, this is exciting moving forward because what it does is it not only democratizes access of data, but it democratizes the sort of number of people that can use this data. And I think that's going to be a bigger and bigger deal as we move forward. So, but I want to stop and take a big step conceptually back. And that is, I think one of the biggest problems we have now in sort of human health and thinking about genomics in human health is that you, me, and everyone on this call is walking around with variants that have an association with disease of 10 to the negative 140, right? But we don't have those diseases, right? Like those are diseases that present in childhood, et cetera, et cetera. We're missing those diseases. And so one thing that I think is necessary is really to understand the cis and trans effects of local genetic mutation. And I think we've been thinking about sort of ethnicity and population genomics in way too broad a way. And it's really underserved, particularly admixed populations, but really at admixture at every level. And for example, I I think when we maybe do a GWAS, we say, oh, okay, well, I'm going to isolate all the German people, right? Well, it's a big mixture of people and so on and so forth, as I'll show you mathematically in a minute. What I'd really like to see is sort of areas between recombination hotspots where we could say, oh, this person is Portuguese 4 on one strand and German 8 on the other strand. Their mutation, the mutation of interest or the variant of interest is in the German 8 haploblock. So we should be doing GWAS in that haploblock and not in the population in general. So we did this other experiment to think about local genetic ancestry, and honestly, we built on this a lot at Carnegie Mellon this week. But it was a very simple experiment. We took people from Puerto Rico in 1,000 genomes, as well as Britons, and then people Han Chinese. What we did is we said, oh, okay, let's just make heat maps, just brute force in the haploblock around TNF alpha and the haploblock around HLA. And we got exactly what you would expect. So around TNF alpha, uh, we got 8 clusters for Puerto Ricans, 5 clusters for Britons, and 3 clusters for Han Chinese. It's one of the most conserved regions of the entire human genome. In a variable region, again, we got what you'd expect. I believe 17 regions for Puerto Ricans, 13 regions for Britons, and 11 regions for Han Chinese in the HLA region. This is just background haploblocks with common SNPs. So that to me is very interesting. It says that with large enough biobank numbers, in diverse populations, we can actually easily look at cis and eventually trans effects. And I just worked with a team led by a woman named Maria Chiquina to really think about how to calculate trans effects as well from these background haploblocks this week. And we hope to publish that in the next couple of months. So really, I mean, what we're interested in, I think with these biobanks, and really what we can do in an actionable way, or thinking about combinations of loci associating with patients and disease, cis and trans effects with haplotypes, and then eventually what we want to do is generate machine learning models for which set of diseases these drugs work on. And we were lucky enough to work with some sort of causal incident inference models earlier. So, I think, of course, then, we're in this massive shift in terms of having more data, but also, we're trying to think about that in the context of having more and more powerful and more accessible deep learning tools. So a couple of years ago, I did a bunch of interviews of large pharma companies and others in terms of AI, and I think a lot of the most pressing issues are really around data in terms of these AI models, and so we ran a sort of a data management for precision medicine scientific conference, which was very well attended. And if you go to this link, you can see that. And I would say that these were a series of roundtables with experts in the field. And even as a commercial company, one thing we've seen a lot of traction on is having roundtables with people that aren't necessarily clients or even officially partners. People want to come see experts speak, and not just one expert, as you all are. And I'm humbled that you're listening to what I'm saying, but groups of experts talking sort of frankly about their fields and what they're thinking. And I think that, I mean, there's a lot of excitement around data, but also we don't have good models for how we're going to credit folks for generating data, and that's a big one. And so, I think one of the things I've been thinking about a lot lately in my sort of day job is, sort of if I have a disease specific TRE, maybe not a maybe not a nationwide biobank, a local data spectrum. I think somebody accidentally pressed the unmute button.
Nala, could you please? Thank you.
So anyway, so I've been thinking a lot about, if I have a disease specific biobank, say for ALS, as well as a general biobank, how can I integrate those, let people run complex models, and then send them out to a bunch of pharma as well as academics. And actually, we've seen uh, a lot more receptivity in terms of pharma, in terms of actually helping academics get data. So that's a happy thing. Um, and then also, sort of pharma playing back into that and really reinforcing those complex models. And I think, this is a place where the entire community is really part of the whole system in terms of, sort of incentivizing folks building clinical diagnostics tools, including sequencing, like we were talking about in the run up to this, really sort of clinical diagnostics, sort of making their subtyping and stratification measures available, and then refining them based on academic and industry research, so that eventually, we can give these to patients. We're not, and I think the objective is not to tell smart clinicians what to do. It's to have indications really for subtypes, and then if clinicians want to try those on additional populations, they should be enabled to do so. But then we should be able to capture that data and look at efficacy. So that's to me has been really exciting, and I mean, I think the UK Biobank and other large biobanks have really enabled all of this. And so the last thing I want to talk about is sort of this philosophy of data generation. I think a lot about AI and other deep learning models, and really the idea that deep learning is much more accessible to people than it was even 2 or 3 years ago. And I mean, um, AI is good at certain things, and particularly in terms of writing code. And I think thinking about Ethan Molik's co-intelligence and so on and so forth. Thinking about alignment with AI is something that's really going to be there in the next 5 years. And thinking about young researchers and trying to get them in their journey, I think that's going to be a critical step. But there's certain things that AI are not good at, and I think that's the biggest thing, is to understand what AI are good at and what they're not good at. And one thing that they're not awesome at is really uh, moving data to information, and then contextualizing that information in knowledge. So, I mean, for example, like, I think it's we ran a sort of an AI competition, and really putting protein interaction networks and other knowledge graphs in at critical stages of graph neural networks was really important in terms of getting reasonable outputs out. We just sort of finished that project yesterday, and I can provide some links if people are interested. But I think that what we really want to do is essentially come up with better Bayesian priors for everything we do. And for that, we're going to need more and more and more data. And we'll probably understand using AI, we'll probably be able to understand and predict what data sets are going to give us better Bayesian priors. So people that are sort of able to produce data quickly from biological samples, I think, will eventually be incentivized to do that. Although, frankly, I don't think we've figured out an incentivization structure for that yet. And so thinking about, I mean, these efficacy models and feeding back into the research framework is, I think that's an early step into being able to think about generating more data. And so I think AI is actually going to push us to generate cleaner and cleaner and more volumes of data. And really, again, I think it's important to think about models that help us generate specific data that we can um, use to then narrow down efficacy. But I think I've shown you some really simple examples of how to sort of predict efficacy from some data with really basic bioinformatics tools. So it doesn't have to be fancy. It just, I think, really takes some thinking by really smart people, and luckily there's lots of smart people, young people out there, particularly young people, who can do amazing things right now. So I'd like to thank you. There's a whole bunch of people that have worked on all kinds of aspects of these projects and so on and so forth. And I know it's been sort of a grab bag of a bunch of different ideas, but hopefully this sort of reinforces the idea that if we have the data out there, we can do sort of simple analyses, and B, that the AI will take those analyses and really turn them into sort of a desire for more and more data. And with that, I'd like to stop and take any questions you may have.
Ben, thank you so much. That was a whirlwind tour of many different subjects as it relates to data. So any questions for Ben? There's nothing in the chat right now, but you can put things in the chat or raise your hand and ask a question. Uh, Ben, I'll start off. So I wrote down a number of questions, but I'll start with the first one I jotted down. When you talked about cod liver oil, it hit home. I had a grandmother that used to make me take it all the time when I was a kid. But is that a good example of what we geneticists tend to acknowledge but ignore to a great extent? And that's the effect of the environment, our diet, our lifestyle, our social standings, all the things that go into all these types of calculations that actually present as our final disease phenotype or non-disease phenotype. How do we incorporate those sorts of information sources into these data systems to make them more accurate, particularly with AI as something that can take in huge amounts of data, but how do we reduce it to something useful?
That's a great question, and something I was fortunate enough to get to think about a lot, over the earlier part of this week. And one example I would give you is that we had a team really looking at PQTLs and really mapping PQTLs onto graph genomes, right? And then they said, "Well, what do we do from here?" And I said, "Well, the thing that we need to look at then is being able to peel the onion. So some of these disease subtypes, there might be copy number variants. Some of them, there's additional SNPs. Some of them are affected, the penetrance is affected by other cis and trans effects from background genome. And finally, there are environmental effects, right? And so the idea is that for any one of these subtypes, we want to, in my opinion, we want to sort of peel back the onion and say, hey, are there obvious proteomic changes? Are there obvious changes in the graph genome, right? Are there, actually, let me hold off on the proteomic one and say, are there obvious changes in the graph genome? Are there obvious GXG changes, right? From background. Are there obvious SNPs in other genes that we can look at? So we peel those back and we say, oh, okay, we've done what we can with the molecular readout, right? And we still aren't seeing what we need. So then we can look at environment. And I think, there are two really good molecular readouts of environment. One is epigenomics and the other one is proteomics. And proteomics can be really amazing looking at a snapshot readout of sort of the integration of both
Mhm.
genomic and environmental factors. And so that's really very exciting. But also, I mean, we now so there's a lot of models for this, and some people go as far as sort of RDF graphs. But I mean, I can say that DNA Nexus is sort of set up as a star graph, but with that star graph, I can look at in any given genetic background, I can see the effects of individuals or groups of um, um, phenotypic assays. So again, I think my hope is that in the next few years, for most major disease types, say type 2 diabetes or Alzheimer's, we'll be able to establish sort of the major molecular definitions. We'll be able to start looking at sort of the phenotypic uh, changes there, and then we'll really be able to start looking at some of the genomically idiopathic diseases and say where can we look for environmental changes? I mean,
Mhm.
if people are exposed to teratogens or whatever, it may not matter for a small group of patients what their genome looks like.
Great. Thank you. Cindy, did you have a question?
I didn't. I just think it's well, I've got lots of questions always for Ben, but I think it's really compelling, this idea of being able to bring the metadata that isn't so sensitive together using these concepts of Dockers, which I just know the word, right, Ben? I just know it's a concept that you talk about, and that, like I said, Melissa Klein has talked about it in a GA4GH working group meeting. That was the first place that I came across it. And I think that we can transfer lessons from some cohorts to other cohorts where there's such privacy challenges. And then I guess I'll also say on top of that, that you've mentioned proteomics. These data are less sensitive, but we're drafting in the heels of genetics, and we need to err on the side of caution with any data, I think, that are adding value to genetics. So thank you so much for the presentation, Ben. I think it's a nice opportunity for this group to see some of the uplifting work that DNA Nexus is doing that isn't necessarily revenue generating directly, but important for the science.
Great.
Yeah, it's been fun to get a lot of people involved. And as many of you know, there's probably 10,000 active users right now of UK Biobank and about 30,000 people that have some sort of access. And so that to me is exciting that there are so many people working on a data set. Another thing I'm really excited about in terms of crossing is, I mean, so now we have basically all of the haploblocks, all of the background haploblocks for the white British population, right? So then thinking about how we can take lessons from that into other populations, like, so we have 100, nearly 100% of those. So then, we have a start on, I don't know, Nigerian or South Asian populations, right? We don't certainly have anywhere near the coverage there, but maybe we have 30% that we can start with that are the same in those populations. And that's probably the next step, is to start thinking about, once we can define haploblocks, thinking about defining haploblocks across populations. There's somebody, Ricky, I think, has come off video mute. Maybe did you have a question? Or you just happened to be off mute?
Just to say hi.
Oh, cool. Hi.
There's one person I know here, I think, Stefan Gruber. And the rest of you, I'm just getting to know. It's
Awesome.
Is that the Stefan Gruber from originally Chicago, Family G, the Lynch syndrome? I don't know if he's on.
I don't know, but hi to Stefan. Regardless if they're watching the recording. Alan Scholdinger is on this call, which is super cool. We've worked in the same institution a couple of times. But anyway, it's nice to see you. I don't know if you wanted to chip in anything, but
yeah, no, I was going to ask you a question then. Really staggering talk, amazing stuff that's going on. My question is directed more towards the implementation of genomic health side of things. So I think, you've made a really compelling argument about how large biobanks, particularly government-led ones that are quite large and linked to, for example, National Health Services data, can be so valuable for discovery. And of course, combining those biobanks make them even more powerful at the end of the day. I could see a very straight shot from, data from UK Biobank vis-a-vis our Future Health, where at the end of the day, those discoveries can move right into patient care in the UK. Thinking about the US and, its fragmented healthcare system and how this is going to roll out in the US. I mean, all of us is fine, but I just don't know that there's a direct line of translation. And it leads me to ask you, what do you think the role of, I'll call them local biobanks are in, small relative to everything else, healthcare systems, hospitals, et cetera. Less useful, I think, for discovery. But how do you see those? What's the value of those, and is there any value of those in making that critical link to implementation of genomic health?
So I guess I would say two things. I mean, I think the one most valuable thing is, I mean, a lot of this again is based on people, right? So at some of these payer provider institutions, you're going to have world experts in a particular disease class. And what I've seen from where I sit is, if you can get a couple of major academic health systems together and put together a critical mass of disease specific genomes, those are extremely powerful things. And I guess the place I've seen them be most effective is in common rare diseases, right? Like chronic pancreatitis or ALS, something like that, right? Where we know that there's a complex genetic environmental interplay, and we know there are subtypes. But if you had 5,000 genomes for a disease like that, that would be amazing. And if you have an expert clinician or practitioner who sort of has enough clout to sort of assemble people at their own institution, and they can get 1,500, 2,000 samples consented for research use, then all of a sudden that's a game changer, right? And now, fortunately, with cloud infrastructure, groups of these people can get together and work together. But I think also we've seen a paradigm shift in pharma, where I think 10, 15 years ago, pharma would go and they would go try to get one of these cohorts and make proprietary data. And that, what I've seen is that be flipped on its head. Now pharma want to support generating relatively open data. They, of course, like to get early access to things, but generate relatively open data and pay for part of it, and make it open to academics, which that's something I've been shocked about. But I think part of that is because, particularly for mid-sized pharma, their research budgets have been cut. So collaborating with academics has been really, I think, early in their mind. In terms of small biobanks per se, I think that is a really underlying the use of Docker. Again, being at academic institutions and them using all of us and using UK Biobank and hopefully more of these major biobanks and crossing them with their internal biobank. I mean, I think to do that kind of federation meaningfully, they're going to be able to want to go and run the same software in their biobank and these other places. And that makes containerized software important. Thanks for the question.
Any other questions? I'm going to ask one more, Ben. You finished up with AI, and something that's not an well, it is an issue with the cohorts, the many cohorts that we have as part of our organization, the IHCC. Not all of them have electronic health records. Many of them do, particularly the large national governmental ones, but many of the small ones do not have electronic health records. Do you think there's an opportunity here to use AI to scan paper records somehow and produce electronic health records? And also, does AI have potential for some of the harmonization difficulties we have when we put together disparate databases with different phenotypic definitions too, to try and harmonize that data?
Well, so it's a great question, and I think I would go back to the idea that, I mean, what we want is alignment of experts with AI, right? And so these are two examples that highlight that, I think, extremely well. If we should have AI doing what AI or models really are good at, and humans should do what humans are good at, right? And so the first question is absolutely, like, as we've seen, there's these big powerful models that are not actually necessarily super good for nucleotide type of things, but for semantic things. I mean, being able to translate those things and cluster them and so on and so forth for semantic output. A lot of our sort of current AI models are amazing at that. And so I think for use case one, that's a place where AI can be leveraged in a relatively straightforward way. But you want somebody driving that AI that understands the data, right? And I think that's the really important thing, is to have a subject matter expert who understands what those records are, right? Who understands how those records were gleaned and so on and so forth. And I think it's very valuable. I know there's a couple of projects looking at patient response data in the UK Biobank, for example. And I think that's awesome. The second part is, there's a cottage industry of people using AI to do harmonization of data sets. And honestly, harmonization is difficult and expensive, and it's great people using AI, but I still think there's no free lunch, right?
Okay.
Like, if you just took a random person and you throw AI at like metadata harmonization,
Yeah.
you're probably going to get garbage in, garbage out. You really want somebody who fundamentally understands, what the ontologies are, but not only that, but what are the ontologies that you want to get out, right? So I have a friend who works for a company called Synthesis. It's like Synthesis without the Y. And they do a lot of ad hoc metadata harmonization for people, right? But he's always saying, he's the CEO of this company, and he goes, I cannot tell you how many meetings I sit in where I just ask people what they want their column headers to be, and they have no idea, right?
Yeah.
So you have to be able to tell whatever computer system you have what you want harmonized to, right? And then, but as like the people here, I think it's really critical to think about, like, well, Okay, so you get something harmonized to something, but then how is that going to play with all the other biobanks in the IHCC Atlas, or at least a significant fraction of them? And that is, I think, what takes coordination is thinking about. Well, I mean, is OMAP a reasonable standard, or like is it OMAP plus kind of thing? You know what I mean? And so I think those are questions, or like Mondo, blah blah blah. So I think those are questions we're going to be asking ourselves for a long time, and we're going to need like a lot of leadership. In terms of that ontological space. And I think Melissa Handel and others are doing brilliant work, but then we need sort of another layer between the Melissa and the Chris Mungles of the world, and like practitioners of of large biobanks. And so I'd encourage you to have them come and speak, and then maybe in conjunction with some people that are doing sort of practical data harmonization as well.
Thanks, that's great. We got about 4 minutes left for any other questions. If anyone has anything they'd like to ask Ben, I know he did put his email in that last slide. So, and he said he'd be happy to continue discussions with people that are interested on things. So if you didn't get anything answered today, he is a great explainer. And he mentioned at the beginning that in a panel, he asked that question, with unlimited funding, what would you do with it, of several people. And I thought it was amazing, Ben. There was a lot of diversity to the answer, but there was a lot of similarities too. Everyone sort of said what we need to do is bring data together that's being produced around the globe to have a more powerful data set to look at than the balkanized, stratified, siloized, if that's a word, system that we have right now.
Yeah. I couldn't agree more. So, well, yeah. And thank all of you for, working on this. Like the data generation is going to be the biggest part of work. If we're going to realize, some of these goals of really understanding, right drug, right patient, right time. So thank you for doing that, everyone.
Thank you.
It all starts with you. So, yeah.
Quick question, and I'm sorry I came on late and I was in clinic, but are you the person to invite to a webinar on how to use the UK Biobank, for instance, on a departmental level for my institute?
I would be,
can
except
follow
I'm
on
switching
and
jobs tomorrow. Ping me on LinkedIn, and I will direct you to somebody who will be amazing.
Please send us your new email address then after you switch jobs.
Absolutely. I'll just put my personal email in here. Anybody can reach me out to me there. It's no big
Perfect.
deal.
And congratulations.
Oh, well, thank you. Yeah. So.
Okay. Latifa, I think you had an announcement about the next webinar you wanted to make before we close.
Oh, yes, yes, thank you, Scott. Thank you, Ben, for the insightful and engaging presentation. And just to let everyone know that we will inform them about the forthcoming webinar very soon. Just stay tuned. And thank you again for everyone participation and for attending today's webinar. Thank you, Scott, also for moderating this session. So stay tuned.
Thank you, everyone. We will be back in a month with another new exciting webinar. I can't tell you what the title is right now, as Latifa said, because we're sort of trying to figure out exactly who's going to be giving it, but that information will be coming out soon. And we love having you here. So thank you very much for participating. Bye, everyone.
Bye.
Bye, everyone, and have a great
Thanks again, Ben.
day.
Bye. Apologies, joining late.
No worries.
Bye, thank you.
If you missed today's webinar session, we will be sending you the recording.
Where is this? Okay, thank you.
You're welcome.
Where is that? I'm looking for a button to leave the meeting. Leave. Yeah.